Okay, I think I'm going to go ahead and get started. Welcome you all to another exciting semester of Genetics Colloquium. Most of you have known me enough years to know that I get particularly excited when we have a colloquium about bacteria. So, yay, we have a few other bacteria enthusiasts out there. So it's my real pleasure today to introduce Tu-Ahn Huynh, who is a candidate to be a genetics trainer. So we have bulk up the bacteriology here in our program. So I can tell you a little bit about Tu-Ahn. She did her undergraduate degree in Australia at the University of New South Wales. And then after that, worked on her graduate work at UC Davis in the lab of Valerie Stewart, who, well, it's hard to come up with exact words to describe that. I'm going to go with a legend of two-component transduction, and a very interesting individual as well. After that, John was to do a postdoc with Joshua Woodward at the University of Washington in Seattle. And that was when she transitioned a bit from two-component transduction to studying what we call a second messenger, and one of those enigmatic little molecules in cells, cyclic di-AMP. We know that it has a tremendous impact on a variety of different phenotypes and a number of different bacteria, but it was really abundantly unclear how that was mediated. And John's work there and what continues, as she joins the Faculty of Food Science here, is an investigation of the role of cyclic di-AMP in the physiology and metabolism of cell wall, and the impacts it has on those very large and diffuse phenotypes of pathogenesis and virulence. So without further ado, I'll turn it over to John. Tell us about the most exciting research with cyclic di-AMP.
Thank you so much, Nicole. So before I start, thank you everyone for the really great questions that you sent me. They're so helpful for our, ongoing projects and our work. And yeah, now I have to live up to your level of intelligence. But anyway, so as Nicole introduced, my lab works on bacterial signal transduction mechanisms, and one major focus right now is on this nucleotide second messenger. Hold on. Called cyclic di-AMP, and it's essential for many bacteria to grow to infect. And the stories that I'm going to tell you today are about how such an essential molecule has to be used in moderation by bacteria to avoid toxicity, to get things just right. Okay. I'm not able to move the slides. Hold on. They were moving before. Wasn't there a Zoom thing?
It could be. Let's see. Yeah, maybe what was going on. Maybe just hit escape. I think that might do it. Yeah. Or more.
Practice with Zoom.
No. Okay, so we're actually in Zoom now. But why is this?
Yeah, how do we exit?
Oh, there we go. I think we're back. We're back in business.
All right. For some reason, that's called troubleshooting, everyone. Okay, so so my lab work primarily on bacteria, and personally, I've been working with bacteria forever, and I think they're like fascinating because they're so adaptable. They can be found in every corner of the planet. They can be found in extreme environments, and so they're great to understand, diversity and adaptation and signal transduction. So one bacterium that we know and love in the lab, mostly because we are good with its genetic tools, but Listeria monocytogenes is a highly versatile bacterium. is a notorious foodborne pathogen that has been associated with many foodborne outbreaks. So you may have heard of it that way. But this is a picture, false colored picture of Listeria. And Listeria is actually a soil bacterium originally, and it's quite abundant in natural environments. So it easily infects agricultural animals. Infected animals and shed Listeria, and so Listeria can easily contaminate the human food production chain. And what gives Listeria a huge competitive advantage is that it grows under conditions that exclude other pathogens like high salt, low pH, refrigeration, etc. And then Listeria can, of course, infect humans, and it doesn't just stay in the gastrointestinal tract. It can spread out to systemic infection sites and causes lethality in immunocompromised people. In the field of vector pathogenesis, Listeria has been studied extensively as an intracellular pathogen. What that means is that it can invade and replicate inside the cytosol of mammalian cells. And this is quite unique compared to things like Staph aureus, for instance. So this is a macrophage cell infected with Listeria. Listeria is fluorescently labeled in red. And you can see it happily moving around, polymerizing host actin to protrude itself. And it grows in macrophages just as fast as it does in lab media. And as it does that, it elicits a number of immune responses. And one of the immune responses that was known for a long time to be elicited by Listeria was type 1 interferon response. This is an antiviral response, so by in itself is quite an interesting topic as for why bacteria induce this response and what this response does in bacterial infection. So that's a story for another day. So that was known for a while, and discovered that the molecule that induces type 1 interferon response is cyclic AMP, and that was discovered by Josh Woodward. That's my postdoc advisor. And this was very exciting at the time because cyclic AMP had never been known to be produced by a living organism. So this is the structure of cyclic AMP. It's basically two ATPs that are cyclized by two phosphodiester bonds. And the enzyme that synthesizes cyclic AMP is DAC, or diadenylate cyclase. Now, Listeria was the first bacterium known to make cyclic AMP, but by bioinformatics and experiments, we now know that most bacteria make cyclic AMP, and they range from environmental bacteria, such as cyanobacteria, to human pathogens, such as Mycobacterium tuberculosis. And many archaeal species make cyclic AMP as well, although experimentally it's not quite as well studied. So, and actually, well, not every species needs cyclic AMP to grow, but many do, especially those in the Firmicutes phylum, such as Bacillus, Clostridium, Staphylococcus, Listeria. Bacteroides is a gram-negative class of bacteria in the gut, and they're known to require cyclic AMP for growth as well. And we know the cyclic AMP is essential because the DAC gene is essential. If you delete DAC in these species, it's a lethal deletion. Now, we can't delete that, but we can genetically deplete it by putting an inducible promoter so that you can reduce its expression. And I'm going to use data from Listeria as an example as for how DAC is essential. So we characterized, our postdoc lab characterized the DAC depleted mutant, and we found that it is diminished for growth in macrophages. This is a macrophage growth curve, so you can see wild type growing by more than 1 log of CFU at the end, and the DAC mutant didn't grow at all. In the mouse model of infection, here are mice infected with Listeria for 2 days. We can also assess bacterial burdens as an indicator of virulence. And the DAC mutant is down by 3 to 4 log CFU, so that's a significant attenuation. And so much progress in the field has been made to understand why cyclic DNP is essential in some bacteria, especially in gram-positive bacteria. But what's also becoming increasingly evident, but much less understood, is that cyclic DNP accumulation is toxic as well. And one way to regulate cyclic DNP levels is to degrade it. And there are two phosphodiesterases that degrade cyclic DNP, PDEA and PGPH. They have different catalytic domains. We have generated a mutant that's deleted for both, and I'll refer to that as PDE, short for phosphodiesterase. We can measure cyclic DNP levels by mass spectrometry. So you can see here that the PDE mutant has 3 or 4 times more cyclic DNP than wild type because it doesn't degrade it. PDE grows fine and blood, but it's very attenuated during infection. And this is, again, a mouse infection experiment where we found PDE to be down by 3 or 4 log CFU in burdens as well. And this is almost actually as attenuated as the DAC mutant. So it turns out that cyclic DNP homeostasis is critical for virulence. So you might ask, okay, well, we can deplete DAC to reduce cyclic DNP, and we can block hydrolysis to increase cyclic DNP, but do those levels ever occur in real life? And yes, cyclic DNP levels are dynamic in wild type bacteria as well. So this is Listeria growing in just a lab frost medium. So you just can see what a typical Listeria growth curve looks like. And at different phases of this growth, we collected aliquots to quantify cyclic DNP. And just wild type Listeria can accumulate up to 4 times cyclic DNP as it enters early stationary phase. And in the face of stress, such as antibiotic stress, this is a beta-lactam antibiotic that represents a big class of antibiotics in the clinic. So here we expose Listeria, healthy Listeria, to a sublethal amount of concentration of ceftriaxone, which is 1 beta-lactam antibiotic, for half an hour. And within half an hour, Listeria basically, got rid of half of the cyclic DNP levels. This is actually about the same level as the DAC depleted mutant, so it's quite significant. So cyclic DNP levels are dynamic. And what that means is that perhaps we can look to manipulate cyclic DNP levels as a therapeutic sort of strategy. And what's interesting, though, is that it turns out that cyclic DNP toxicity is widespread. So here are some examples of bacteria, but actually, from what I see in the literature, pretty much every bacterium that makes cyclic DNP cannot tolerate it. So these are the bacteria for which the PDE mutants are safe in terms of whether they're stress sensitive or attenuated. Listeria, Bacillus anthracis, both group A and group B strep, Mycobacterium tuberculosis, Borrelia, which is a Lyme disease agent, Streptomyces. What's interesting, though, is that not all of them require cyclic DNP for growth, and so cyclic DNP toxicity is even more widespread than essentiality. So that's appealing because then perhaps we could inhibit cyclic DNP hydrolysis to develop new antibiotics. But we're not quite there yet. Also, my lab is not quite a chemistry lab. So what we're interested in right now is how is cyclic DNP toxic? What are the mechanisms for this? And by doing so, we will learn more about the functions of cyclic DNP in the bacterial cell and infection. And well, hopefully that will inform therapeutic strategies. Okay, so when we found the attenuated virulence phenotype of PDE, The field was still reasonably young at the time, and we didn't know very much about the molecular functions of cyclic AMP. And so we turned into this technique called a phenotype microarray. And what it does is you can subject your bacteria under a large number of culture conditions in 96 well plates. And then you can just, refine phenotypes that are relevant to the process. So in our case, we were looking for phenotypes that could be relevant to infection. Of course, if you screen, you need a readout to screen. And the readout in this case is bacterial respiration. So when bacteria respire, NADH gets oxidized into NAD, the electron gets transferred to a tetrazolium dye. And upon reduction of that dye, it turns purple. So you can read purple, the purple color, as how metabolically active the bacteria are. And then you can, superimpose your experimental responses to a reference response to find your phenotypes. And to be honest, it's quite a messy screen. It's just not that clean. But what we found was one response that we found to be clear cut was an osmotic stress response. And it's messy, and we couldn't really where this is going, whether it's more sensitive or resistant, which we could tell is different. And so we needed to validate it in the lab. And we grew wild type and PDE at increasing sodium chloride. This is a basic osmotic stress thing. And then when comparing relative growth of these strains, you could see that PDE is readily inhibited. Now, sodium chloride is an ionic compound, so we needed to double check if it's, an ionic effect or not. And so we turned to sorbitol, which is a non-ionic compound. We found the same thing, which is that PDE is highly sensitive to osmotic stress in general, not an ionic stress. So then bacteria are under osmotic stress all of the time, whether they be Listeria growing in a salty food or Listeria growing in a very thick, dense cytosol of a macrophage. There's osmotic stress all the time. And when osmotic stress occurs, bacteria start losing turgor. The cells start shrinking. And so one way to counter shrinkage is to import compatible solutes. So these are things that can accumulate inside bacterial cell without harming them. And carnitine is rich in mammals. And Listeria is good at importing carnitine through a transporter called OPUCA. So we went and looked for carnitine transport in wild type and PDE strains. The way we did that was we incubated Listeria with a radio label carnitine substrate, and then we put this whole thing under osmotic stress, and then we counted radioactivity after stress. And what we found here first is that wild type can rapidly import carnitine post-shock. OPUCA is a mutant that is deleted for OPUCA, and of course it doesn't import carnitine. PDE is not quite as abolished, but it's pretty effective for carnitine transport. We had known at the time that cyclic AMP, as a second messenger, it would go on and directly bind proteins and regulate protein functions. And so we went ahead and tested to see if cyclic AMP would bind OPUCA. And luckily it does. This is a binding curve for cyclic AMP at increasing OPUCA concentrations. This is just the cytoplasmic soluble portion of the protein. And we found binding affinity to be good as well within the physiological concentration of cyclic AMP. I'm not showing you here, but OPUCA doesn't bind related nucleotides, so specific. I don't have time to go through data, but over the past years, the field have learned that actually cyclic AMP binds other osmolyte uptake channels, especially potassium uptake. And we verify that in our lab biochemically, as well as by measuring potassium concentrations in the wild type and PDE. And again, PDE is depleted for potassium. And so putting those things together, cyclic di-AMP appears to be good at inhibiting osmolyte transport, in particular carnitine and potassium. So at this point, we know that PDE mean is attenuated. It's also the factor for osmolyte transport. Are those two phenotypes related? Well, I think they are. However, there's a catch, which is that mutants that can't transport these things are just not quite as attenuated as PDE. And so we think that there's more than osmolyte transport that is contributing to bacterial adaptation during infection. So then we turn to another omics approach. We had learned at this point that cyclic di-AMP regulates bacterial metabolism, but not quite at the molecular level. There were, phenotypes here and there that indicate that. So to be systematic, we profiled the PDE and wild type metabolomes. And there were a bunch of things that were different, as you noticed in your questions. And we were then, we became interested in two compounds in particular, D-alanine, D-alanine, and UDP-Myrnac. And not because we're cell wall experts, but because they both map to the same pathway. And that gives us some sort of comfort in our omics data. So this, briefly, this is a pathway that synthesizes precursors for bacterial peptidoglycan. If you don't think about bacteria every day, well, bacteria need a cell wall. A cell wall is like a coat that protects it from things like antibiotics. And peptidoglycan is the major component of the cell wall. So the key precursor for peptidoglycan is UDP-Myrnac pentapeptide, and it's made by UDP-linked intermediates, including UDP-Myrnac, as well as alanine derivatives, including D-alanyl, D-alanine. And we found both of these compounds to be significantly down in the PDE strain by total untargeted metabolomics. So then we went ahead and quantified these compounds by tandem mass spectrometry, and we were able to quantify them all, but decided to focus our mechanistic analysis on this part because the reactions here are more defined to us. So here we quantified glycine in wild type and PD strains just as a control. And then walking through the reactions here, L-alanine and D-alanine levels were similar in wild type and PDE, so that's fine so far. But then the products, D-alanine, D-alanine, as well as UDP-Myrnac penta, are significantly down in the PDE strain. So then we decided to chase down on DDL, which is the enzyme, which is essential, that ligates two D-alanines into the dipeptide, D-alanyl, D-alanine. And again, I don't have time to go through all the biochemical data, but mostly to summarize the finding from our paper, which is that we knew that cyclic di-AMP inhibits potassium. And it turns out from our enzyme assays that DDL needs potassium to work. And so we think that that's one physiologically relevant way for cyclic di-AMP to inhibit DDL function. We also found cyclic di-AMP to directly inhibit DDL as well, though the inhibition is a lot more modest. And so, combining these two things together, basically at high cyclic di-AMP, bacteria just don't quite make as much peptidoglycan precursors. So then would that have any consequence on the cell wall, which is of course, the target of many antibiotics in the clinic. So we went ahead and quantified those things. First, we quantified peptidoglycan and cell wall. And indeed, the PDE strain has only 75% of the cell wall compared to wild type. And that turned out to be sufficient to sensitize PDE to a broad range of cell wall stresses. So I'm going to show you two examples. One is beta-lactam antibiotics, such as cefuroxime. These antibiotics inhibit peptidoglycan synthesis in bacteria, and they're also the primary antibiotics to treat human Listeria infection. And we found the PDE strain to be highly sensitive to cefuroxime and other beta-lactams as well. These are relative growth rates of wild type and PDE increasing cefuroxime concentrations. Another cell wall stress that we tested was lysozyme. And this is interesting. So mammals abundantly produce lysozyme. During bacterial infection, lysozyme chews up the glycnac beta glycosidic bond, and so lysozyme is not good for the bugs. And part of Listeria's success as a pathogen is that is evolved to become quite resistant to lysozyme, and yet the PDE strain is highly sensitive. Okay, so far what we had learned is that high cyclic DIMP inhibits peptidoglycan synthesis that results in a loss of cell wall integrity. Part of the mechanism we found was via inhibition of DDL. Now, what turned out was that DDL overexpression was just not sufficient to restore cell wall defects. And so at this point, we had to turn to forward genetics to map out the different defects that PDE has. So just to recap, PDE is sensitive to beta-lactams such as cefuroxime. And so we treated PDE with a chemical called EMS, which induces point mutations in the genome. From there, we were able to isolate cefuroxime-resistant mutants, and we selected for those that are restored to wild-type level, as well as retain high cyclic DIMP. And then we tested them for the ability to infect mammalian cells. This is a plaque assay. Briefly, the way it works is that when Listeria infects mammalian cells, it can spread from one cell to the next. And by doing so, it lyses cells, leaving plaques. And plaque sizes are, for some reason, anecdotally well correlated with mouse virulence phenotypes. And so we use this routinely in the lab as a quick readout for virulence. What's interesting is that of all the suppressor mutants that we selected that met all criteria, they're all recovered for infection as well. And we're right now in the process of trying to understand how these mutations restore cell wall as well as infection. But what we think is that cell wall defects are a major contributor to virulence attenuation at high cyclic DIMP levels. So it's important to have a good cell wall when bacteria infect. Okay, so cyclic DIMP regulates many proteins. These proteins have different functions, so cyclic DIMP regulates many pathways. And you might wonder, okay, how do we disentangle all of these, interconnected web? And so we turned to another omics thing. This time we did RNA-seq for PDE and wild type. And what became apparent right away was that in the PDE strain, there was this whole cluster of genes that was down by at least one order of magnitude in PDE. And what that is the entire Listeria pathogenicity island, which is comprised of a transcription factor called PERF-A. And PERF-A upregulates a whole suite of virulence genes that are required for Listeria to infect and to spread from one cell to the next. So then we validated our RNA-seq data by qPCR. We chose just two genes within the PERF-A regulon for this. HLY is an early gene because it has a perfect PERF-A box in the promoter. FA is a late gene because the PERF-A binding box is a bit more divergent. Okay, so PDE is down for the expression of both HLY and FA compared to wild type by at least tenfold. What I'm showing you though is only the basal level of virulence gene expression. when in fact, there's a lot of glutathione in the mammalian host. And PERF-A is activated by glutathione, and so that basically upregulates the entire virulence gene, set of genes. And so we revisited our gene expression experiment by growing Listeria in glutathione to see what happens. As we would expect, wild type strongly upregulated both HLY and ACTA. PDE does that as well, but nowhere near the wild type level. So then, so this is because the way the mechanisms by which PERF-A is activated by glutathione are quite well understood. So we decided to look into how Listeria metabolizes glutathione, which is not quite as known. And there are two ways for Listeria to get glutathione. One is to take it from mammalian hosts, and the transporters are not known yet. And the other is to synthesize it via a glutathione synthase, GSHF. So now we looked into glutathione synthesis simply by growing Listeria in a defined medium that doesn't have glutathione. And in the lab, we can measure glutathione. And we found PDE to be much less capable of glutathione synthesis than wild type. To look into glutathione uptake, we deleted GSHF from both the wild type and PDE strains, and then we grew them in a medium with glutathione. So then we found that in this system, the PDE strain is also highly deficient for glutathione uptake. So putting these two, stories together, this is a skeleton picture of how we think cyclic DIMP is regulating bacterial adaptation during infection. One is that it can regulate the virulence program through glutathione metabolism. Right now we're looking into identifying glutathione transporters and understanding how cyclic DIMP is inhibiting GSHF. Another way for cyclic DIMP to regulate pathogenesis is through controlling cell wall homeostasis, which is, of course, important for bacterial success during infection. Okay, so that's cyclic DIMP. The lab is really cyclic DIMP, obsessed at the moment. However, what happens to the product of cyclic DIMP? Well, cyclic DIMP is degraded into PAPA, which basically is just the linear version of cyclic DIMP, if you wonder. And we found that in Listeria, PAPA has to be degraded further. And the enzyme that does that is called nRNA in Listeria. So we did what we're good at. We prepped an RNA enzyme, we did enzyme assays. This is a standard Michaelis-Menten kinetics curve for an RNA degrading PAPA, and it degrades pretty well. So this is a simplified version of the data. We did test other substrates as well, and RNA can degrade cyclic DIMP if you give it enough time and a lot of cyclic DIMP, but the clear preference is for PAPA. We can also measure PAPA in the bacterial cells. Here, compared to wild type, the delta nRNA mutant, which is deleted for nRNA, accumulates by, I think, 20-fold more nRNA than PAPA than wild type. We can recover that by complementing the mutant with a wild type version of nRNA, but not with a catalytically dead version of nRNA. And so the effect here is totally due to the hydrolysis of PAPA. Interestingly, PAPA accumulation also attenuates virulence. So again, this is a plaque assay compared to wild type. We got the nRNA mutant has just about half of the plaque size, which we need to do a real mouse infection experiment for this, but by experience, I would expect about 3 log CFU down. And that phenotype can be recovered by a wild type nRNA allele, but not a catalytically dead nRNA allele. And so again, the defect here appears to be related to PAPA hydrolysis. Okay, and so that's basically all the stories that I'd like to tell you today. I need to thank, of course, my lab. Chaya is our 4th year NDTP student, and she's been working on the PERF-A project. Our former lab technicians, Stephen Massa and Jed Godfrey, worked on the peptidoglycan project, and they're both in grad schools right now. We have a long-term collaboration with Bill Gutile at the University of Missouri Pharmacy School. He is an expert on quantifying peptidoglycan precursors, which is not trivial because they're pretty unstable. We talk with a lot of labs on campus because, as you can imagine, Cyclic IMP does a lot of things, and we're not necessarily expert at everything, at least not in the beginning. And so we really appreciate everyone's help on campus, especially JD Sauer, who helps us with the immunology aspect of Listeria, and Joe Dillard, who helps us with cell wall biology and funding. And yeah, thank you so much. Yeah.
So at the last slide, you showed the PAPA. That PAPA has a specific kind of pathway to contact with toxic, or just activation of the PAPA that check feedback to change the amount of cyclic that I get. What do you do?
Yeah. Thank you so much. That's what we, at least I was thinking about for a while. Okay, so one thing, as you mentioned, can PA feedback into select IMP metabolism, because sometimes substrates can feedback and inhibit the enzyme. We actually did measure select IMP in the delta mRNA mutant, and that's the same. And so it doesn't appear in the case of Listeria that PAPA has much to do, does much to intervene with select IMP metabolism. So one tidbit that I left out because of time was that PAPA is also a product of RNA degradation. So during transcription, abortive transcription, mRNAs have to be degraded, and eventually they make their way into short oligonucleotides such as PAPA. So my guess is that most of PAPA in Listeria perhaps comes from that source. That's why it doesn't quite integrate with IMP. So what can PAPA be doing? We don't know for Listeria. We really try to get suppressor mutants, and so far none of them turned out to be real yet, as in they couldn't be validated. In E. coli and Pseudomonas, which are gram-negative bacteria, it has been shown that PAPA can misprime promoters and globally shift transcription. I don't know if that could be the case for Listeria because the nRNA mutant by itself grows fine in lab media. If there was a global transcriptional effect, we would expect it to be sick, and that's not the case. Yeah, thank you. Yeah, I'm fascinated with PAPA and fascinated with what it could be doing. Perhaps we might be looking at another second messenger. Yeah.
The sexual organism, something related to the pathway you're already looking at, or something else altogether that makes the difference?
Yeah, I don't know. I would love to find out. I think there are two bacteria for which the mechanisms were essentially unknown: Listeria and Bacillus subtilis. For Bacillus subtilis, it appears to be related to potassium, because people. So there's a group in Germany that works on Bacillus a lot, and turns out that they can knock out, delete the DAC gene if they also get rid of potassium in the medium. So it appears that cyclic DIMP inhibits potassium transport. If you don't have cyclic DIMP, there is so much potassium coming in that the cells burst. Doesn't appear to be that way for Listeria because the DAC mutant seems to be still sick when potassium isn't there, but it appears to be related to another pathway called the stringent response in bacteria. So when bacteria are nutriently starved, they start making an hormone called PPGPP. And sorry, it's another thing, but PPGPP tells bacteria to be, you know, now it's time to make more amino acids, etc. However, PPGPP can also inhibit growth. So the gist of the story is that at low cyclic DIMP, Listeria starts making more PPGPP, and that's the false signal to stop growth. We don't know why this occurs. Yeah, we don't know. So it's pretty complicated. Yeah.
I think you answered my question already, but I'll just ask a question. Is there a relationship between cyclic AMP and dicyclic AMP? And I think the stringent response probably is the question.
Yeah, I can tell you that Listeria doesn't make cyclic AMP. I really tried. Couldn't find it by mass spec or by or identify the gene. So perhaps not. In Listeria. And I think Firmicute bacteria such as Bacillus, Cerdulus, and Staphylococcus aureus don't appear to make cyclic AMP either. Yeah. But I mean, just because we couldn't detect it doesn't mean it could be produced in some conditions that we don't know of.
But you said, like does make it.
E. coli makes cyclic AMP and not cyclic di-AMP. It does, yeah. So yeah, PAPA. My thinking is that most of PAPA comes from RNA decoration because there is just about 4 or 5 micromolar of cyclic di-AMP in the cell. It's not a whole lot. Yeah.
Thanks for your comment. You know that removing unreceptional structural binding motif?
No, unfortunately, no. Yeah, I wish. No. So thus far, we haven't found it yet. And even if you look at so quite a few cyclic DIMP proteins have been solved, like in terms of crystal structures, and there is just not a whole lot of commonality in terms of binding site because cyclic DIMP itself can take different conformations. Sometimes it's a U-shaped and sometimes it's straight when it binds. I mean, yes, if you want to bind a phosphate, you need some sort of positively charged residues like lysine, histidine, etc. Still, we haven't found a motif to let us predict a motif. Yeah, but perhaps we're just not a informatically capable lab. Yeah.
Oh, that's why me asking if anybody else wants to ask me questions. Well, great. If you don't have any further questions, let's thank Joy again.
Thank you all for the super great questions. And yeah, if anything comes up or if there's more feedback, I would love to hear.